Hello, everyone. Today, we will discuss an interesting approach to tackle antibiotic resistance. We will explore how computer-aided drug discovery, or CADD, is used to find new ways to fight antibiotic resistance. My name is Yori Yuliandra. For the next couple of minutes, I'm going to be presenting my study with the title, Fighting Fosfomycin Resistance in Silico Screening for FosA Inhibitors. Antimicrobial resistance continues to be a serious threat. It is projected that AMR can cause 10 million deaths per year by 2050, taking more lives than cancer. The World Health Organization has released a list of critical priority pathogens which can cause severe and deadly infections. For example, multi-drug-resistant Pseudomonas aeruginosa. Unfortunately, the current antibiotic discovery model is not delivering sufficient agents to combat present levels of antibiotic resistance, leading to fears of the arrival of post-antibiotic era. One of the antibiotics struggling with resistance is fosfomycin. It is actually quite an ideal antibiotic, which has broad-spectrum activity. In addition, it also has good solubility and low toxicity. One main mechanism for its resistance is the enzymatic inactivation by FosA, an enzyme produced by bacteria. Previous studies reported that the homologs of FosA are present in the genomes of many bacteria. For example, in 98.8% of Pseudomonas aeruginosa genomes. With that being said, FosA is a promising and novel target for the development of antibacterial agents. This is because inhibiting this enzyme will potentiate the activity of fosfomycin. The good news is the crystal structures of FosA are available in the protein data bank. Also, the proteins are co-crystallized in complex with a novel inhibitor. However, this inhibitor is a new chemical entity which can take a lengthy process of up to 10 or 15 years to be approved as a drug. Meanwhile, antibiotic resistance is speeding up at an alarming rate. All right, so the main objective of the study is to discover potential inhibitors of FosA to be used in combination with fosfomycin as adjuvants, but we can break it down into 3 stages. The first one is to perform in silico study by using the crystal structures of FosA and the co-crystallized ligand, any one. This work is done through computer-aided drug discovery. Secondly, to conduct in vitro antibacterial testing of the fosfomycin adjuvant combinations. And lastly, to further design and synthesize novel FosA inhibitors. Today's presentation will cover only the first stage of the study. In the molecular modeling, we combined structure-based drug design and ligand-based drug design through molecular docking and pharmacophore screening for potential adjuvants of fosfomycin. 2 crystal structures of the enzyme were used for the modeling. The ligand libraries were taken from various databases, including DrugBank and FDA, with the total numbers of more than 500,000 ligands. In our molecular docking simulations, we used the binding site of any one as the receptor for docking. To improve the accuracy and reliability of our results, we employed a progressive refinement strategy from high-throughput virtual screening, proceeded to standard precision, and finalized in extra-precision docking. Our baseline for this refinement was docking scores of fosfomycin. As previously mentioned, the crystal structures of FosA used in this study are co-crystallized in complex with an inhibitor called any1, as seen here. The molecular features of this inhibitor were used as pharmacophore model to screen against ligand databases. The results from this stage will be combined with those from the molecular docking studies performed earlier. So from the in silico work that we have conducted, we come to the hit list of the computational simulations. Actually, we have a big list of clinically approved drugs that have been identified as potential fosfomycin adjuvants. We can probably group some of those drugs into their classes, as we can see on this slide. For further in vitro testing, we have made some selections of potential candidates as highlighted in this list, representing adjuvants from both antimicrobials on the left-hand side and non-antimicrobials on the right-hand side. For example, succinyl sulfathiazole, kefonicid, and adavovir from antimicrobials, and losartan, prednisolone, and troxarutin from non-antimicrobial classes. Some of these drugs available on the market will be purchased and some others will be synthesized in the lab. So now we will proceed to the next stage of the study, which is to evaluate the antibacterial activity of the selected candidates in combination with fosfomycin against bacterial strains that are resistant to fosfomycin. Should these drugs prove to be potent FosA inhibitors, this novel approach could reduce the lengthy and challenging regulatory approval process and the developmental cost associated with new chemical entities. These fosfomycin adjuvant combinations can be a crucial reinforcement in the battle against antimicrobial resistance and to save lives. Thanks for your attention, and we'd like to acknowledge Australia Awards for the PhD scholarships received by Yori Yuliandra during this study, and to Drug Discovery Initiatives, the University of Sydney, Australia, for the funding of the study. We'd also like to thank our external collaborators from Emory University and University of Pittsburgh, USA. Again, thank you.